Доброго ранку, шановні колеги. Сьогодні у нас 11 жовтня 2023 рік, 10:00. Дозвольте розпочати засідання. Відповідно до рішення вченої ради Сумського національного аграрного університету протокол 19 від 28.06.23 року та наказу по Сумському національному аграрному університету 410К 07.08.23 року про затвердження складу разової спеціалізованої вченої ради у Сумському національному аграрному університеті створено спеціалізовану вчену раду для прийняття до розгляду та проведення разового захисту дисертації Хе Сунтао на тему комплексний екологічний, фізіологічний та генетичний аналіз реагування представників роду кукурбіта на сольовий стрес. Complex ecological, physiological and genetic analysis of the response of cucurbita genes representative to cell stress. На здобуття ступеня доктора філософії у галузі знань 10 природничі науки за спеціальністю 101 екологія. Згідно з даними карти присутності, в нас є присутні, тому пропоную зареєструватися.
Дякую.
У нас є присутній інцидент, я підтверджую вашу участь у засіданні, що ви опонентів, що ви приймаєте участь. Так, все, у нас відповідно до процедури. Голова ради Коваленко Ігор Миколаївич, доктор біологічних наук, професор кафедри екології та ботаніки, проректор з науково-педагогічної та навчальної роботи Сумського національного аграрного університету Міністерства освіти і науки України, присутній в аудиторії. Рецензенти: Жатова Галина Олексіївна, кандидат сільськогосподарських наук, професор кафедри екології та ботаніки Сумського національного аграрного університету Міністерства освіти і науки України, присутня. Бондарєва Людмила Миколаївна, кандидат біологічних наук, доцент кафедри екології та ботаніки Сумського національного аграрного університету, Міністерства освіти і науки України, присутня також. Опоненти: Лихолад Юрій Васильович, доктор біологічних наук, професор, завідувач кафедри фізіології та інтродукції рослин, Дніпровський національний університет імені Олеся Гончара, Міністерство освіти і науки України. Бачимо.
Присутній.
Дякуємо. Писаренко Павло Вікторович, доктор сільськогосподарських наук, професор, завідувач кафедри екології, збалансованого природокористування та захисту довкілля, Полтавський державний аграрний університет, Міністерство освіти і науки України. Раді бачити.
Присутні, взаємно.
Прошу, ми зареєструвалися. Тобто на підставі реєстраційної картки присутні 5 членів спеціалізованої вченої ради із 5 затвердженим рішенням вченої ради Сумського національного аграрного університету, протокол номер 19 від 28.06.23 року та наказом по Сумському національному аграрному університету номер 410К від 7.08.23 року. Кворум необхідний для засідання є, і ми всі маємо офіційні підстави для того, щоб розпочати засідання. Також здобувач є, науковий керівник також є. На порядку денного питання захисту дисертації аспіранта денної форми навчання кафедри екології та ботаніки Сумського національного аграрного університету Хе Сунтао на здобуття ступеня доктора філософії з галузі знань 10 природничі науки за спеціальністю 101 екологія. Є присутні. Тема дисертації: комплексний, екологічний, фізіологічний та генетичний аналіз реагування представників роду кукурбіта на сольовий стрес. Complex ecological, physiological and genetic analysis of the response of cucurbita genus representative to sun stress. Роботу виконано в Сумському національному аграрному університеті. Науковий керівник, доктор біологічних наук, професор, завідувач кафедри екології та ботаніки Сумського національного аграрного університету Міністерства освіти і науки України, Скляр Вікторія Георгіївна, також присутня. До спеціалізованої вченої ради, шановні колеги, надійшли такі документи. Ось тут папка з документами є. Заява щодо проведення атестації здобувача. Копія першої сторінки паспорта громадянина Китайської Народної Республіки. Копія диплома магістра. витяг з наказу про зарахування до аспірантури, засвідчений в встановленому порядку, академічна довідка про виконання відповідної освітньо-наукової програми номер 38-2022, висновок наукового керівника, доктора біологічних наук професора Скляр Вікторії Григорівни, висновок про наукову новизну, теоретичне та практичне значення результатів дисертації. Дисертація у друкованому і електронному вигляді є. Відбитки копій наукових публікацій, зарахованих за темою дисертації, засвідчених у встановленому порядку. Шановні колеги, автор всіх документів ми перевірили, підтверджують, що ці всі документи є присутні на захисті. Усі документи відповідають встановленим вимогам, дисертація до захисту подається вперше. Чи будуть, колеги, зауваження чи пропозиції щодо порядку денного?
Немає.
Тому вношу пропозицію, хто за те, щоб затвердити відповідний озвучений порядок денний. Хто за? Прошу проголосувати. Хто проти, утримався. Дякую за одноголосне рішення. Рухаємося далі. Дозвольте зачитати біографічні дані аспіранта. Хе Сунтао, 1980 року народження, громадянин Китайської Народної Республіки, освіта вища. У 2010 році закінчив магістратуру у Хенанському університеті за спеціальністю генетика та селекція рослин. Працює в Хенанському інституті науки та технологій з 2011 року по теперішній час. Навчання в аспірантурі Сумського національного аграрного університету з 6 листопада 2018 року по 6 листопада 2022 року. Тема та індивідуальний план наукової роботи здобувача Хе Сунтао були затверджені рішенням вченої ради Сумського національного аграрного університету протокол номер 7 від 26 грудня 2018 року та протокол номер 4 від 3 жовтня 2022 року. 28 грудня 2022 року була проведена публічна презентація за апробації дисертації на кафедрі екології та ботаніки, де роботу було рекомендовано до розгляду та захисту у разовій спеціалізованій вченій раді. Здобувач Хе Сунтао виконав освітню наукову програму 6 листопада 2022 року за спеціальністю 101 екологія та отримав відповідну академічну довідку номер 54-2022. Науковий керівник, доктор біологічних наук, професор Скляр Вікторія Григорівна. Основні результати і положення досліджень відображено у 12 наукових працях. У 6 статтях 3 статті видано у провідних наукових фахових виданнях України, 3 у виданнях, віднесених до міжнародних баз даних Scopus та Web of Science, і 6 тезах доповідей на наукових конференціях. Матеріали статей і тез відображають основні положення і висновки дисертаційної роботи. Наукові публікації відповідають вимогам пунктів 8 і 9 порядку присудження ступеня доктора філософії та скасування рішення разової спеціалізованої вченої ради закладу вищої освіти наукової установи по присудженню ступеня доктора філософії. Відповідна постанова Кабінету міністрів номер 44 від 12 січня 2022 року. Дисертація відповідає вимогам Міністерства освіти і науки України номер 40 від 12.01.2017 року про затвердження вимог до оформлення дисертацій та порядку присудження ступеня доктора філософії та скасування рішення разової спеціалізованої вченої ради закладу вищої освіти наукової установи про присудження ступеня доктора філософії. Відповідна постанова 44 від 12 січня 2022 року. Шановні колеги, чи є питання стосовно поданих здобувачем документів? Немає? Дякую. Хе Сунтао, you have question? No? Окей. Слово надається здобувачу для викладення основних положень дисертаційної роботи. За погодженням із здобувачем захист буде відбуватися відповідно англійською мовою. Хе Сунтао, would you start to present the main result of your research?
Okay.
Okay.
Dear Chairman and the members of the Special Scientific Council, allow me to present the results of research on the topic of the dissertation. Note about 20% of the world's arable lands are subject to salinization. According to FAO, the total area of land affected by salinization exceeds 928 million hectares. At the same time, increasingly important subsets aimed at elucidating the mechanism of response and adaptation of plant organisms to salt stress are becoming more and more important. These studies should first of all cover the leading agriculture groups, include representative of the family capability, which are widely cultivated in the world due to its economic, medicinal, agricultural, and ecological value. Considering the above, the issue of clarifying the ecological, physiological, genetic aspects of the response to salt stress of responsive of the cucurbitaceae family and the genes cucurbita, the family of their salt resistance becomes relevant and managing. This treat on the basis of the use of both classical and modified scientific and technical means. The aims of the research was to establish mechanism of response to salt stress and adaptation to it, which are implemented by representative of the genus cucurbita and different leaves of organization, as well as eco-physiological aspects of the formations of salt resistance, quantitative and qualitative characteristics of plants when using grafting technology to achieve the set goal. Following tasks were formulated. Tasks are presented on slides. The scientific novelty for the first time based on the results of observation due the entire growing season. The influence of salt stress on a complex of quantitative and qualitative cosmological characteristics of plant. Plants were studied, the features and the regulation of the accumulation of ions of mineral substance in the vegetative and generative organs of pumpkin and concentration of salt stress have been established. Physiological and biochemical transformations that appear in pumpkins against the background of salt stress were comprehensively investigated and clarified. For the first time, the eco-physiological interactions and transformations that occur during the cultivation of cucurbitaceae using grafting technology and the use of a vascular modifier fungi were investigated and clarified. Research methods. The work was performed based on the application, theoretical analysis, synthesis, explanation, generalization, mathematical and statistical and empirical, experiment, measurement, comparison, research methods. In study aimed at establishing the effect of salt stress on the morphological characteristic, variety and degree of damage to pumpkin plants, as well as on the exchange of organic and inorganic compounds. The flow of the leading physiological processes. The following experimental scheme was used: control, water 0 mol nickel, low salt stress, 6 mol nickel, high salt stress, 120 ml alcohol. Pumpkins were two varieties were in the study, Yanjin and Miben. The influence of salt stress on the morpho structure, habit, and vitality of packing plants was studied based on studies carried out throughout its ontogeny. First, seed work germinated and seeding was obtained. Packing plants at a different stage of research. At the beginning of research, the study plants did not have statistically significant difference in the value of morphoparameters. After nickel treatment, there was a statistically significant decrease in the set of plants and the value of leading morphoparameter, leaf sulfur air, dry weight, steam lung. With the increase of salt concentration, the inhibition degree of plants increased gradually. Against the background of salt stress, the plants also showed negative quantitative signs, yellowing, cleaning of leaf, loss of ability to form generative organs, and as a result to form fruits. The results of the vitality analysis based on these morphometrics showed that against the background of an increase in the concentration of salt, there is a decrease in the level of plant vitality. This is confirmed by the data presented in the depot. With the increase of nickel concentration and level of salt stress, the salt damage index and the salt damage rate of the two varieties of packing showed an increasing trend in the engine variety and the salt stress. Damage symptoms appeared later, and the level of damage was significantly lower. That is, according to the set of these characters, the engine variety compared to the medium variety demonstrate higher salt stress resistance. According to the results of the study of the influence of salt stress on physiological processes and characteristics, it was established that in Parkinson's against the background of an increasing in nickel concentration, the indicators of photosynthetic rates, transpiration rate, water utilization ratio decrease. Against the background of increased salt stress, there is also a decrease in stomatal conductance, stomatal limit, and an increase in intercellular CO2 concentration. Against the background of increasing salt stress, the amount of chlorophyll in the leaves of packing plants increased. 2. Against the background of salt stress, the content of MDA also increases. 3. Against the background of salt stress and an increase in nickel concentration, an increase in the content of protein was registered. Study varieties of Parkinson's. 4. Against the background of salt stress and an increase in the concentration of nickel in the studied variety of Parkinson's, established in the variety engine a tendency to increase the content of soluble sugar was resistant. All the given facts prove that transformation related to the exchange of organic compounds play an important role in the complex of processes of response and adaptation to salt stress in Parkinson's. The effect of salt stress on air exchange were studied. It is established that the NAR content in plant organs of both varieties increase. Plants of the engine variety mainly accumulate NAR in roots, and of the medium variety in the airway organs. The accumulation of K as was also studied. maintaining hierarchy in the above ground can help reduce the damage from excessive accumulation to plants. K-NA ratio can better reflect this effect. The accumulation of CAs was also studied. The accumulation of MGs was also studied. Based on the results of summarizing the obtained data, it was found that against the background of source stress caused by high nickel concentration in plants of both varieties of pumpkins, the absorption of nutrients decreased. The obtained data indicate that an important reason for the difference in the salt tolerance of the two investigated varieties of pumpkin is the difference between them in the content of NA in different organs, which is manifested against the background of source stress caused by different concentration of nickel. In the process of genetic research, salt resistance was construction of pumpkin WQA transcription factor phylogenetic tree. It was divided into 3 groups according to the compensation of the conserved domain, and the second group was further divided into 5 groups. Modern analysis of the pumpkin WQA transcription factor family, it can be seen from the figure that all WQA transcription factors are homologically 1, indicating that these moduli constitute homologous WQA domains. Conserve the motif of pumpkin WQA protein, the amino acid length of the 4 motifs reached from 11 to 38. Analysis of WKG expression under salt stress. Optimization of experimental conditions for GFP detection. Optimization procedure of transient transformation. The interactions that occur between the sand and the salt-resistant pumpkin root stock when using the technology of grafting in the cultivation of pumpkins were studied. The features are also established sugar cruise and total sugar accumulation in flesh of grafted root watermelon lines and on root watermelon lines during fruit development stage. It is established that the use of a vascular mycorrhizal fungi in the cultivation of melon crops based on the grafting technology leads to a change in plant metabolism. Fruit mice accumulation and different treatments during the fruit development stage. The materials of the dissertation work are used in the educational process of the Department of Ecology and Botany of Sumy National Agrarian University when teaching such disciplines as ecological physiology of plants, agroecology, biology. The main results position thesis was considered and discussed under 6th Conference of different rank. According to the materials, 12 scientific papers were published, including 3 articles published in a scientific journal, included in the database of scopes, Web of Science, and 3 articles published in professional scientific journals of Ukraine. The conclusions of the research results were announced during the report. Allow me not to read them.
Okay, thank you, He Sунтао, for your report. The report is finished. Let's go on our meeting, our colleagues, dear members of council. Are there any questions? Please ask some.
Okay,
Galina Zhatova, professor,
thank you.
your questions.
My question is, why do you think that the difference in spatial temporal expression patterns of the 12 WRTY transcription factors are related to their functional diversity on the salt stress? Thank you.
Okay. During plant evolution, plants have developed a set of adaptive mechanisms to adapt to environmental change and various biotic and abiotic stress. Under stress conditions, plants not only represent a quantity to short-term stimuli from internal and external environmental signals, but also activate a series of protective systems to cope with long-term stress, change in spatial temporal expression patterns of transcription factors play a key role in regulating plant response to stress. Tissue-specific expression analysis of the 12WKY target transcription factors revealed typical tissue-specific expression of most genes. Normally, gene expression localization is compatible with its function. Therefore, it is speculated that the difference in spatial temporal expression patterns of these WQA transcription factors may be related to their functional diversity under salt stress. Over.
Yes, thank you. Thank you. And the next question.
One more question, please.
Okay. As a part of your research, of your study, you carried out a transient transformation. I'd like to know what is the difference between transient transformation and stable transformation? Thank you.
And transient transformation and stable transformation are both techniques used in molecular biology to introduce foreign deoxyribonucleic acid into host cells for various purposes. However, there are significant differences between the two processes. transformation involves the temporal introduction and the transformation of foreign deoxyribonucleic acid in host cells. The foreign deoxyribonucleic acid is not simply integrated into the host cell's genome and does not get passed on onto subsequent generations. Transient transformation is usually used for short-term study, protein production of or functional analysis of genes or protein. Stable transformation refers to the permanent integration of foreign deoxyribonucleic acid into the host cell's genome. The introduced deoxyribonucleic acid becomes a stable part of the host cell's stable materials and can be passed on the subsequent generations during cell division or in sexually reproducing organisms. It allows for long-term transformation and inheritance of the desired included by the introduced deoxyribonucleic acid. Over.
Thank you for your question, Nomo.
No, okay. Людмила Бондарєв, PhD, please, your questions.
Okay, thank you. My first question is, the sweet substance in watermelon are sucrose, fructose, and glucose. Why did you study only glucose? And what is the reason for the higher sugar content of triploid plants compared with diploid plants?
In different stages of watermelon fruit development, the content of fructose and glucose change little, and the content of sucrose was higher. Therefore, sucrose is mainly studied in this study. In the future, it is also necessary to study the change and mechanisms of fructose and glucose after chromosome W. The original gene function remains the original function, and the gene may be selected or mutated to other functions. The reason why the sugar content of triploid watermelon is higher than that of diploid watermelon may be that the gene regulating sucrose synthesis retains the original function after W and chosen. The ability of sucrose synthesis. Over.
Okay. More questions, please.
Yes, one more question. Please explain in detail. Why does young Gen pumpkin improve his salt tolerance by delaying the increase in MDA content?
Okay. Under salt stress, the plant plasma membrane is the first to be damaged, leading to the destruction of the selective permeability of the plasma membrane. That is, the permeability becomes larger, and many organic functions are destroyed. Melon dihydrohydra has been accepted as an indicator of the level of lipid peroxidation, the stronger the lipid peroxidation, the higher the malondialdehyde content, and the greater the membrane's permeability. Therefore, it is generally believed that the amount of malondialdehyde accumulation and the increase of membrane permeability are negatively correlated with the salt tolerance of varieties. There is the more malondialdehyde accumulation, the greater the increase of membrane permeability, and the weaker the salt tolerance. The amount of malondialdehyde accumulation and the increase of membrane permeability can be used to compare the surface side of varieties. In addition, malondialdehyde affects the absorption and accumulation of ions by cells through affecting membrane permeability and membrane transport protein. Over.
All right, thanks.
Yes, I'm satisfied with your answer. Thank you.
Okay, you continue. Yuri Nikolaev, Doctor of Biological Science, Professor, please your questions.
Yes, I have two questions. Please
Okay.
detail, why does reduce photosynthesis in pumpkin?
Okay. And salt stress can reduce plant photosynthesis by affecting CO2 diffusion to binding sites and changing the structure and function of organelles responsible for light reaction, and changing the chemical process of dark reaction, and inhibiting the transfer of assimilation products. The results show that salt stress could decrease the stomatal conductance and transpiration rate of pumpkin leaves, which was beneficial to reduce the water loss in the plant and enhance its resistance to salt stress. The decrease of stomatal conductance will cause the transport of CO2 required for photosynthesis to mesophyll cell to be blocked. But due to the decrease of plant net photosynthetic rate caused by salt stress, the CO2 required for photosynthesis is also reduced, so that CO2 accumulation in plant cell and in intercellular CO2 concentration value increase. Over.
Thank you. The importance of your work for agriculture.
Okay. Pumpkins have developed root system, vigorous grounds, strong water and fertilizer absorption capacity, strong disease resistance, strong growth adaptability, easy management, and good plant benefits, and are planted over the world. And pumpkin not only as a traditional Halloween decoration, it can be used as a vegetable but also as food. In recent years, with the deepening of scientific research, the edible and health care value of pumpkin has been recognized by people. Pumpkin fruits is rich in beta carotene, crude fiber, and a variety of trace elements, which has a high edible value. At the same time, pumpkin also has the medicine effects of lowering blood sugar, detoxifying, and eliminating carcinogens. In the papers, the salt tolerance of the selected experimental material engine pumpkin was analyzed from the physiological and ecological, biochemical, and molecular levels, and it was found that engine pumpkin has a high salt tolerance ability. So today, the
Хе Сунтао, stop. Next question. All good.
okay.
Please, Professor, you will have next question, please.
Да. I'm satisfied.
You have, okay. Pavlo Pisarenko, Doctor of Agricultural Science, Professor, please your questions.
Dear Хе Сунтао, please specify what are the effects of pumpkin rootstock on watermelon science?
Okay, the rootstock can improve the ability of drought resistance, cold resistance, heavy metal pollution resistance, and pest resistance of sands. It can change the leaf color, yield, and quality of sand. Pumpkin rootstock can improve the yield and desert resistance of watermelon soil, sand, and also changes the flavor of watermelon, which has a certain influence on the quality of watermelon. For example, the sucrose contained in this study. Over.
Thank you. And one question. What is the use of Yanjing pumpkins in production?
First of all, Yanjing pumpkins can be grown in saline alkali land to improve land utilization. Secondly, it can be used as a rootstock from the perspective of being used as rootstock in production. The root of rootstock traps most of the nut, reduce the transport of nut to the organs of the shoot, and makes the leaves free from air poisons to the greatest extent. Therefore, the accumulation of salt ions in the root is more beneficial to sand than that in the shoot.
Okay?
Thank you.
Okay, thank you. Hhe Sунтао, I have a question, one question, please. Please explain in detail, how does the morphological structure of pumpkins change under salt stress?
After nickel treatment, the leaves of pumpkin plants became smaller, plate hair became shorter, and the plant growth was significantly inhibited. With the increase of salt concentration in the treatment, the degree of plant inhibition gradually increased. After 60 more nickel treatment, the growth of the plant was slowed down and the leaves was bent. Head part of leaf tree tipped. Leaf margins were yellow and the leaf were significantly smaller, while the leaf of Yanjin are normally slightly smaller, and the concentration of 120 more. The growth of the plant was further delayed. Most of the leaf tip and leaf mandate were yellow, showing above obvious systems of salt damage. The growth was seriously abnormal. The leaves were seriously curled and appeared to fall or even die, and could not normally flower and bear fruit. Yanjin leaves further become smaller with part of leaf tip, leaf edge sculpture, but can flower normally and bear fruit. Over.
Okay, thanks. Dear colleagues, no more question? No? Thank you. Dear He Sунтао, close please your presentation. Close presentation. Questions finished. Our questions, no. Please. Okay, thank you. So, let me familiarize everyone with the content of the consultation about the sciences, knowledge, theoretical and practical signification of the result of the dissertation. Висновок про наукову новизну теоретичне та практичне значення результатів дисертації є Хе Сунтао, комплексний екологічний, фізіологічний та генетичний аналіз реагування представників роду кукурудзіта на сольовий стрес, що подано на здобуття ступеня доктора філософії в галузі знань 10 природничої науки зі спеціальністю 101 екологія. Засолення ґрунту є характерною ознакою багатьох природних екосистем. Воно охоплює близько 20% водних земель планети і є одним із чинників, що обмежують сталий розвиток і модернізацію світового сільського господарства. Тому все більшої значущості набувають дослідження, спрямовані на з'ясування механізмів реагування та адаптації рослинних організмів до сольового стресу. Насамперед вони мають охоплювати провідні сільськогосподарські культури, включаючи представників родини кукурбітації, які широко культивуються у світі завдяки своїм господарським, лікувальним, харчовим та екологічним властивостям. Гарбузові не є галафітами, більшість з них чутливі до сольового стресу і зазвичай демонструють очевидне пригнічення росту в умовах, коли вміст натрію хлора у ґрунтовому розчині досягає 0,1%. Враховуючи зазначене актуальності, набуває питання засування еколого-фізіологічних, генетичних аспектів реагування на сольовий стрес, представників родини кукурбітації, власне роду кукурбіта, формування їхньої солестійкості та управління цією ознакою на основі використання як класичних, так і новітніх наукових технічних засобів. Врахуванням зазначеного метою роботи було визначено встановити механізми реагування на сольовий стрес та адаптації до нього, які реалізуються представниками роду кукурбіта на різних рівнях організації, а також еколого-фізіологічні аспекти формування солестійкості, кількісних та якісних характеристик рослин при використанні технології щеплення. Дисертаційна робота Хе Сунтао виконана відповідно до планів роботи кафедри екології та ботаніки Сумського національного аграрного університету та згідно планів роботи науково-дослідного інституту Хенанського науково-технічного університету. Зокрема, частина досліджень була підтримана грантами ключових дослідницьких та рекламних проєктів провінції Хенань. Наукова новизна роботи полягає в тому, що вперше за результатами спостережень протягом всього вегетаційного періоду досліджено вплив сольового стресу на комплекс кількісних та якісних морфологічних ознак рослин гарбуза. Встановлено особливості та закономірності накопичення іонів мінеральних речовин у вегетативних і генеративних органах гарбуза в умовах сольового стресу. Всебічно досліджено та з'ясовано фізіолого-біохімічні перетворення, які відбуваються в гарбузах під дією сольового стресу. Відібрано 12 транскрипційних факторів родини у гарбуза у відповідь на сольовий стрес. І детально проаналізовано їхні філогенетичні зв'язки, просторово-часові моделі експресії тканинно-специфічні характеристики експресії та транскрипційну активність під дією сольового стресу. Перше досліджено та уточнено екофізіологічні взаємодії та трансформації, що відбуваються при вирощуванні представників роду кукурбіта і родини кукурбітації за технологією живцювання та використання арбускулярних мікоризних грибів. При цьому вдосконалено методичні аспекти оцінки впливу сольового стресу протягом всього циклу росту гарбуза. Та вперше використано методику віталітетного аналізу для оцінки стану рослин гарбуза, а також розроблено систему тимчасової трансформації для вивчення функцій і генів і субклітинної локалізації білка в гарбузі. В роботі набули подальшого розвитку теоретичні та практичні засади використання солостійких підщеп гарбуза для використання масштабних культур. Робота Хе Сунтао має суттєве наукове, теоретичне та практичне значення. Її результати вже використовувалися в навчальному процесі кафедри екології та ботаніки Сумського національного аграрного університету при викладанні, зокрема, дисциплін екологічна, фізіологія, агроекологія, біологія. За результатами дослідження впливу стресу, викликаного різними концентраціями натрію хлору, Хе Сунтао встановлено характер реакції морфологічних ознак гарбуза на сольовий стрес. Показано, що на тлі підвищення концентрації солей відбувається зменшення розмірів і життєвості рослин та прояв у них низки вегетативних якісних ознак, пожовтіння листів, їхнє скручування тощо. Встановлено, що зміни розмірних показників рослин на тлі сольового стресу можуть виступати інформативним індикатором при дослідженні механізму солостійкості та виведення солостійких сортів гарбуза. В дисертаційній роботі показано, що рослини реагують на зростання концентрації сольового розчину, статистично значущим підвищенням величин індексу сольового ураження та ступеню сольового ураження. Результати досліджень засвідчили, що індекс сольового ураження, як морфологічний індекс оцінки стійкості рослин до солі, є простим, легким, точним та достовірним показникам до прямого виявлення ступеня стійкості гарбуза до сольового стресу. Доведено, що сольовий стрес впливає на показники та ознаки, пов'язані з перебігом фотосинтезу та водообміну гарбуза, що призводить до зниження значень чистої швидкості фотосинтезу, розміру продихів, продихової провідності, швидкості транспірації та коефіцієнта використання води, а також до зміни змісту хлорофілу, переважно у бік його збільшення. При цьому зниження показників, що визначають величину втрат води, є чинниками підвищення стійкості рослин до сольового стресу, а показників, що визначають інтенсивність фотосинтезу, чинниками уповільнення синтезу вуглеводів і гальмування росту рослин. Ступінь варіації вмісту хлорофілу у листках на тлі сольового стресу був пов'язаний з сортом рослин, різновидом солі та її концентрації. У гарбуза за сольового стресу зареєстровано підвищено вмісту малонного діальдегіду та проникнення мембранових структур клітин внаслідок їхнього перекисного окиснення. Показано, що у гарбуза перетворення, пов'язані з обміном вуглеводів та амінокислот, відіграють важливу роль у комплексі процесів відповіді та питання до сольового стресу. Зі збільшенням концентрації солі збільшується зміст проліну та розчинних цукрів. Розчинний цукор і пролін є важливими осмотичними речовинами, які регулюють осмотичний потенціал і водний потенціал рослин в умовах сольового стресу. Після семиденного сольового стресу, обумовленого впливом натрію хлор, вміст проліну в листках двох проростків гарбуза був достовірно вищим, ніж в контролі. Встановлено, що сольовий стрес впливає на абсолютні та відносні показники накопичення іонів мінеральних речовин, а також їхній розподіл органами рослин. За цими характеристиками іони натрій, калію, кальцію та магнію продемонстрували високу індивідуальність, що також є результатом і свідченням їхньої специфічної ролі як в аспекті забезпечення реакції, так і адаптації рослин гарбуза до сольового стресу. З'ясовано, що у реагуванні та адаптації до сольового стресу в гарбузі на всіх рівнях організації на тлі вище викладених загальних тенденцій чітко проявляються сортові особливості. Зокрема, встановлено, що сорт яньчжень переважно накопичує натрій в кореневій системі, а мібен у стеблі. Це має наслідком формування відмінностей між сортами і у накопиченні калій, кальцій і магній, а також відмінностей у значенні співвідношення калій-натрій. Загалом за комплексом фізіологічних та морфометричних ознак яньчжень демонструє вищу стійкість до солі. У процесі досліджень проведено ідентифікацію та аналіз експресії родин генів гарбуза в умовах сольового стресу. Відкрито 12 транскрипційних факторів родини у відповідь на сольовий стрес. Детально проаналізовано їхні філогенетичні зв'язки, просторово-часові моделі експресії, тканинно-специфічні характеристики експресії та транскрипційну активність під сольовим стресом. Для вдосконалення та розширення досліджень, спрямованих на виявлення генетичних аспектів формування самостійності, розроблено ефективну систему транзиторної трансформації для вивчення функції генів та субклітинної локалізації білків. Вивчено взаємодію, яка виникає між прищепою та солостійкою підщепою гатруза при застосуванні технології щеплення при вирощуванні масштабних культур. З'ясовано, що та доведено вплив солостійких підщеп на метаболізм підщеп. Показано доцільність використання цієї технології для підвищення стійкості рослин до сольового стресу, підвищення врожайності культур та отримання високоякісної продукції. Показано, що використання арбускулярних мікоризних грибів масштабних культур на основі технології щеплення призводить до зміни метаболізму, впливає на якість одержаної продукції та сприяє підвищенню стійкості рослин до несприятливих чинників довкілля. Був проведений аналіз звіту перевірки дисертаційної роботи Хе Сунтао на плагіат та наявність текстових запозичень. Програма «Страйк плагіаризм», результати якого засвідчили високий рівень оригінальної роботи. Результати перевірки є в комісії. Рецензенти дійшли висновку, що дисертаційна робота Хе Сунтао на тему комплексний екологічний, фізіологічний та генетичний аналіз реагування представників групи кукурбіта на сольовий стрес є результатом самостійних досліджень здобувача і не містить елементів плагіату та запозичень згідно постанови Кабінету міністрів від 12.01.22 року, пункт 44, номер 44, пункт 9. Використані ідеї, результати і тексти інших авторів мають посилання на відповідні джерела. Матеріали дисертації пройшли апробацію на 6 наукових конференціях різного рівня. Вони детально висвітлені в 12 наукових працях, 3 статті опубліковані в наукових журналах, що входять до науково-метричних баз Scopus та Web of Science, 3 статті представлені у фахових наукових журналах України категорії Б, 6 публікацій репрезентовано в матеріалах наукових конференцій. Особистий внесок здобувача підтверджується нормативними документами та науковими публікаціями. Мова дисертації англійська. Технічне оформлення дисертації відповідає загально прийнятим вимогам. Загалом представлена дисертаційна робота відповідає спеціальності 101 екологія та профілю ради. Дисертаційна робота Хе Сунтао за темою комплексний екологічний, фізіологічний та генетичний аналіз реагування представників роду кукурбіта на сольовий стрес, яка подана до захисту у спеціалізовану вчену раду на здобуття ступеня доктора філософії у галузі знань 10 природничої науки за спеціальністю 101 екологія, за своїми актуальністю, науково-теоретичним рівнем, основними результатами обґрунтованості, основними положеннями і результатами опублікованих у фахових виданнях, новизна постановки та практичним значенням відповідає вимогам наказу Міністерства освіти і науки України номер 40 від 12 січня 2017 року про затвердження вимог до оформлення дисертації та постанові Кабінету міністрів України від 12 січня 2022 року номер 44 про затвердження порядку присудження ступеня доктора філософії та скасування рішення разової спеціалізованої вченої ради закладу вищої освіти наукової установи про присудження ступеня доктора філософії із змінами, внесеними згідно з постановами Кабінету міністрів номер 341 від 21.03.22 року. Зміст дисертації повністю відповідає паспорту спеціальності. Головуюча засіданні, кандидат біологічних наук, доцент Катерина Керейчук. Окей. Dear colleagues, according to the procedure, we proceed to the consideration of the dissertation. I invite official reviewer, please.
Thank you very much. A review of the official review for the dissertation of He Sунтао, complex ecological, physiological, and genetic analysis of the response of cucurbita genus representative to salt stress. Submitted by Dr. Field of Study 10 Natural Science, Specialty 101 Ecology. Of the birds, arable lands are subject to salinization. As a background of global climate changes, the area is constantly expanding. Soil salinization leads to up to 50% yield. As a result, salinization becomes one of the powerful factors limiting the sustainable development and modernization of world agriculture. Studies aimed at elucidating the mechanism of response and adaptation of plant organisms to salt stress are becoming more and more important. The research covers the agricultural crops, representatives of the Cucurbitaceae family, which are widely cultivated in the world due to its economic, medicinal, edible, and ecological values. Cucurbitaceae are not halophytes, most of them are sensitive to salt stress. The issue of clarifying the ecological, physiological, genetic aspects of the response to salt stress of representatives of the Cucurbitaceae family and the genus Cucurbita, the formation of their salt resistance becomes relevant, and managing this trait on the basis of the use of both classical and modern scientific and technical means. Research has significant theoretical and practical significance because it allows to deepen knowledge about the essence of the life phenomena, about the role of different levels of the organization in ensuring plant response to the influence of internal and external factors, to determine the possibility and approaches of managing adaptation process, and on this basis, for areas that have undergone salinization, to develop conceptual principles for the formation of sustainable and productive agrocenosis with the participation of the Cucurbitaceae family, including its typical representative, genus Cucurbita pumpkin. Connection of the research with scientific programs, plans, and topics. The research work was carried out according to the task of scientific novelty of the obtained result. For the first time, the influence of salt stress on the complex of quantitative and qualitative morphological characteristics of pumpkin plants was studied. The features and regularities of the accumulation of ions of mineral substances in the vegetative and generative organs of pumpkins under conditions of salt stress have been established. Physiological and biochemical transformations that appear in pumpkins against the background of salt stress were comprehensively investigated and clarified. There have been screened 12 WRKY family transcription factors in pumpkin in response to salt stress and analyzed their phylogenetic relationships, spatiotemporal expression patterns, tissue-specific expression characteristics, and transcriptional activities under salt stress in detail. For the first time, the ecophysiological interactions and transformations that occur during the cultivation of Cucurbitaceae using grafting technology and the use of a basket mycorrhizal fungi were investigated and clarified. The methodological aspects of assessment of salt stress impact during ontogenesis of pumpkin have been improved, and the technique of vitality analysis was used to assess the condition of pumpkin plants. The efficient trends and transformations for the study of gene function and protein substrate localization in pumpkin were developed. The further development of theoretical and practical principles of the use of salt-tolerant pumpkin rootstock for cultivation of watermelon have been gained. The practical significance of the results. The materials of the dissertation were covered in scientific publications and included in the curricula of specialty of 101 ecology at the Summa National Agrarian University, subject ecological physiology of plants, agroecology, and biology. The main results obtained personally by the author. The dissertation provides a theoretical generalization and a solution to the scientific problem of stabilizing the impact of stress factors of Cucurbita pattern. The applicant conducted complex studies on the influence of salt stress on the morphostructure and health of pumpkin individuals, as well as their vitality. The impact of salt stress on indicators related to the photosynthesis process and water exchange, the peculiarities and regularities of the metabolism of main organic substances in the pumpkin plant were established. It has found out the variety of differences between Janssen and Malden cultivars in response to salt stress and the adaptation to it at all levels of the organization. In particular, it was established that the Janssen mainly accumulates sodium ions in the root system, while the Malden mainly accumulates them in the stem. The WRK gene family and analyzed its expression in response to salt stress in pumpkin was identified. For the first time, analysis of the expression of the WRKY gene family in pumpkin under salt stress conditions was carried out, and WRKY family transcription factors in response to salt stress were screened and analyzed their phylogenetic relationships, spatiotemporal expression patterns, tissue-specific expression characteristics, and transcriptional activities in detail. Characteristics of mycorrhizal fungi used in the cultivation of melon crops based on grafting technology, a change in plant metabolism, the quality of the obtained products, and decrease in their resistance to adverse environmental factors were determined. An analysis of the plagiarism check report for the presence of spectrum borrowings strike plagiarism program was carried out, which proved that He Sунтао's thesis complex ecological and genetic analysis of the response of cuckoo represented to salt stress is the result of independent research of the applicant and doesn't contain elements of plagiarism and borrowings in accordance with the resolution of the Cabinet Ministry of Ukraine dated 12 January 2022, number 44, paragraph 9. The ideas, result, and text of other courses are linked to the corresponding source. Number of scientific publications. The main item of the thesis are presented in 12 scientific works. 3 articles were published in the scientific journals, including its database of Scopus, Web of Science. 3 articles were published in professional scientific journals of Ukraine. 6 abstracts of reports of international scientific and practical conferences. Remarks on this content. Positively evaluated He Sунтао dissertation work, its important scientific, theoretical, and practical significance, and relevance of research, it is appropriate to make the following comments and wishes. The first, when studying the changes that occur in pumpkin plants under salt stress during the growing season, it was appropriate use the ontogenesis periodization scale of BBCH. The second, it is desirable to give a more detailed description of the Janssen and Malden varieties. The third, it would be appropriate to present the research result not only in one chapter 3, but in several chapters according to the direction of the research. For example, genetic aspects of adaptation and response of pumpkin to salt stress, ecophysiological aspects of use mycorrhizal fungi, and so on. The fourth, there are inaccuracies in the design of the work. For example, words table, appendix, according to the requirements, are placed in the right corner, but not in the line with the title. Lack of points in the title, and so on. Responded to the dissertation to the specialty and the profile of the specialized academic council. Dissertation by psychological, physiological, and visual academic council for the degree of Doctor of Philosophy in the field of study 10 Nature Sciences, specialty 101 Ecology, in terms of relevance, scientific and theoretical level, main results of validity, provisions, and results published in professional publications, novelty of the formulation and practical significance meets the requirements of the Order Ministry of Education and Science of Ukraine, number 40, January 12, 2017, on approval of requirements for the preparation of dissertation and a resolution of the Cabinet of Ministers of Ukraine of 12th January 2022, number 44, on approval of the procedure for awarding the degree of Doctor of Philosophy and cancellation of the decision of the one-time Specialized Academic Council of the Institution of Higher Education, a scientific institution, on awarding the degree of Doctor of Philosophy with the changes introduced in accordance with the resolution of the Cabinet of Ministers, number 341, dated 21st of March 2022. He Sungtao deserves to be awarded the scientific degree of Doctor of Philosophy in the specialty of 101 Ecology. Official review with PhD Agricultural Sciences, Professor of the Department of Ecology and Botanica of Summa National Agrarian University, Galina Zhartova. Stamp and signature here. Thank you very much.
He Sungtao, you have the opportunity to answer remarks of reviewer, please.
Okay. We sincerely thank Halina Leksenan for reviewing the paper and its critical and thorough analysis. We agree with the compounds provided and shortcomings of the work. Taking into account the comments provided, I should note that the Yanjin is a hybrid variety developed by the Peking Research Group of Henan Institute of Science and Technology, which former variety resources across the country and purified it by cell crossing for 5 years. Maybe is a common commercial variety purchased from market. We will definitely take into account the remarks on the expediency of using the BBSH secure site in general. Such covered the entire ontogeny of pumpkin plants. The period from the very first stages response in this scale to the very last. As for the structure of the work, I should note that it was formed in accordance with the general guidelines and recommendation. However, of course, the formation of the structure taking into the account your comments would contribute to a better presentation of the results of our research, which was of a complex nature. We will also pay attention to this in our future research activity. We would like to thank Halina Leksenan and once again for reviewing the paper and its critical and thorough analysis.
Okay, thank you. Thank
Okay,
you.
thanks. We continue. I invite to next official reviewer, Wonderful, please.
Dear members of Special Academic Council, let me introduce my official review for the dissertation of He Sungtao. There is my full review on the SNAU website, so let me focus on the main provisions and main points. First of all, I must admit the relevance and presented scientific research. Nowadays, global environmental process affect all ecosystems. Agroecosystems are not exception. So the study of salt resistance of the main agricultural crops is a very important scientific problem. As I see, the work was done according to the plan of the research of Department of Ecology and Botany now, and it was supported by grants from the Henan University. An analysis of the plagiarism showed that dissertation does not contain elements of plagiarism. I agree with the reviewer that the dissertation research was scientific novelty and practical significance. Especially, I would like to say that the results of the research are used in the teaching of subject such as ecological physiology of plants, agroecology, and biology. Their submitted dissertation conducts research on the mechanisms of response to salt stress and adaptation to each plants of genus Cucurbita at different levels of their organization. At the molecular, cellular, metabolism levels, at the individual and agro-population levels, at the interspecies and biocynotic levels. This is a very wide range of research. With the generally positive assessment of the He Sунтао's dissertation, I have several appropriate comments and remarks. Regarding statistical processing and presentation of the data. In my opinion, for the most representative presentation of the data, it would be advisable to apply not only analysis of variance, but also regression analysis. It would allow for a better understanding of the overall response of each variety to the influence on factors. I mean about histograms on page number 68, 69, 83, 84, and page 70. Regarding formatting, in the dissertation, some figures 3.8, 3.9, 3.10 consist of several components A, B, C. In my opinion, it would be better to split each of them into 3 separate figures. Representing the contents of ions in the root, stem, and leaf, but these remarks are at the author's discretion. And I see some technical errors, for example, on page number 70, 75, and 96. These remarks do not reduce the scientific level and practical significance of the research, but primarily concern to design of the work and the presentation of the data. In general, dissertation by He Sунтао, complex ecology analysis of the response of Cucurbita genus representatives to salt stress is scientifically relevant, has a high scientific and theoretical level. The main results were published in professional publication. The dissertation has scientific novelty and practical significance and meets the requirements of the above-mentioned order of Ministry of Education and Science of Ukraine, and the author, Хе Сунтао, deserves to be awarded the scientific degree of Doctor of Philosophy in the field of study 10, Natural Science, specialty 101, Ecology.
Thank you. Hhe Sунтао, you have the opportunity to answer remarks of reviewer, please.
Okay. Dear Limina Mekolet-Lana, we thank you for the detailed analysis of our work. We agree with all your content and suggestion. You remark about the need to analyze the results using regression. Analysis is particularly valuable to us. We will definitely use it in our future research. Regression analysis is indeed an informative tool that will constantly allow us to analyze in much more detail the nature of the response of presentative of the genus Cucurbita to salt stress and to review new aspects and patterns in this regulation. We thank Limina Limina Mekolet-Lana once again for reviewing the paper and its critical and thorough analysis.
Okay, thank you. Okay, we continue. I invite an official opponent to speak. Professor Юрий Васильевич Михалат, please.
Thank you. Review the official opponent for PhD thesis of He Suntao, complex ecological, physiological, and genesis analysis of the response of cucurbita genus representatives to soil stress. Submitted for a Doctor Philosophy degree PhD, field study 10, natural science, specialty 101, ecology. In today's world, we are witnessing an escalation of a number of social and environmental problems at both the regional and global level. The negative effects of climate change are becoming increasingly noticeable with various manifestations. These include an increase in the area where soil salinization has occurred. Under such conditions, the issues of the clarifying the features and patterns of response to soil stress, economically valuation crops, especially those that are not calophytes, and for which even a slight increase in the soil contents in the salt in a powerful stress become especially important. This demonstrates the relevance of the topic of He Suntao's dissertation research. This was intended to establish a mechanism of response to soil stress and adaptation to it, which are implemented by representatives of the genus Cucurbita at different levels of organization, as well as ecophysiological aspects of the formation of salt resistance, U-entative and Q-entative characteristics of plant when using grafting technology. To achieve this goal, it was formulate 11 logical, scientifically based texts. The author has studied the interaction that occur between the cyan and the salt-resistant pumpkin rootstock when using the technology of grafting in the cultivation of gorse were studied. By autumn, received the future development of the theoretical and practical principles of the use of salt-tolerant pumpkin rootstock for cultivation of watermelon have been gained. The materials of the dissertation were covered in scientific publication and included in the curricula of the specialties of 101 ecology at the Sumy National Agrarian University subjects, ecological physiology of plants, agroecology, biology, which is confirmed by a certificate. The experimental part of the dissertation is informatively illustrated has a detailed visualization. The main items of the thesis are presented in 12 scientific works. The works consist in introductions, 3 chapters, conclusions, the list of sources, and 5 appendices. The materials of the dissertation are presented in 182 pages. The content of the dissertation fully reflects the main result given in the research. The dissertation has a high theoretical level and its results are significant scientific value for future research. The style of the dissertation meets generally accepted requirements in ecological research. The dissertation totally responds of the field of knowledge 10, natural science, specialty 101 ecology. Discussion clauses and remarks of the dissertation. Positively evaluating He Suntao's dissertation works its important scientific, theoretical, and practical significance, novelty, and relevance of research. It is appropriate to make the following remark and which, taking into account that the work was carried out on basis on international cooperation between Sumy National Agrarian University and Handan University of Science and Technology, the paper should have paid attention to the comparison of soil salinity of Ukraine and China, and the relevance of the issue of adaptation of biological, ecological systems and production processes to this phenomenon of these two countries. In chapter 2, pages 51, it was necessary they describe in more detail the algorithm for calculating the vitality analysis used the assess the vitality of plants against to salt stress. Some of the data presented on figure 3.2, pages 68-69, figure 8, pages 83-84, figure 3.9, pages 85-86, figure 3.10, pages 88-89 should have been present in the form of tables. In the conclusion, it was necessary to detail the content of paragraphs 6 to a greater extent. The work was carried out which extensive as the manifestation statistics methods, in particular analysis of variance. However, the result of these conclusions should have been presented in more detailed form in appendices. The work contains some grammatical errors and shortcomings related for the formatting of the work, for example, incorrect abbreviations. As a suggestion for an applicant, we should note that the work present a number of developments that's important from a practical point of view. So in the future, it is worth paying more attention to the implementation of the result of this research, in particular those relate to the technology of cuttings and all the issues of arbuscular mycorrhizal fungi in farms that the melons. The experiments is also valuable for Ukraine. General conclusion: the dissertation work of the He Sунтао complex ecological, physiological, and genetic analysis of the response of decurbita genus representatives to salt stress, which are submitted for defense to the specialized academic council of the obtaining the degree of Doctor of Philosophy in the field of knowledge 10 natural science in the specialty 101 ecology in terms of relevance, scientific and theoretical level, main results of validity provinces and the result published in the professional publication, novelty of the formulation and practical significance in the requirements of the Ministry of Education and Science of Ukraine, number 40, January 12, 2017, on approval of requirements for registration, dissertation, and the resolution of the Cabinet of Ministers Ukraine on 12, 2022, number 44, on approval of the procedure for awarding of the degree of Doctor of Philosophy and cancellation of the decision of the one-time Specialized Academic Council of the Institution of Higher Education, Scientific Institution on awarding of degree of Doctor of Philosophy with changes introduced in accordance with resolution of the Cabinet of Ministers number 344 date 21st of the March 2022. Based on the mentioned above, I believe that the author of the dissertation, He Suntao, deserves to be awarded the scientific degree of Doctor of Philosophy of the specialty of 101 ecology of the field of knowledge, 10 natural science. Official opponent Yuri Leholat. Thanks.
Thank you, Professor. He Suntao, you are given a possibility to comment the remarks that have been noted in the reviewer of the official opponent. Please, He Suntao.
Okay. Dear Mr. Nicolat, we sincerely thank you for the detailed analysis of our work, given that you are a well-known expert in the field of ecological, physiological, and plant introduction. All your comments are very important for us. Regarding the first content, I should note that the problem of the spread of saline soil and the adaptation of plant and production processes to this phenomenon and its consequence is important for both Ukraine and China. However, for Ukraine, the figures on the prevalence of saline soil vary greatly. According to the Cedar cadastre, saline soil in Ukraine cover an area of over 1.92 million hectares. Of these, 1.71 million hectares are used in agriculture. According to the fall, there are 6.6 million hectares of saline soil in Ukraine, including about 7% of the land under agricultural cultivation. However, in both cases, there are very significant areas that demonstrate the development of this problem for Ukraine. And now, in Ukraine, the issue of plants adaptation to salinity and agriculture on saline soil is receiving increasing attention. China, along with Australia, Canada, the United States of America, and India, is among the saline soil, according to more than 100 million hectares of land in China are affected by soil damage, accumulating for about 11% of the country's land area. The spread of saline soil is largely due to Chinese arid climate. On the coast of Yellow Sea, soil salinity depends on the proximity to the sea. The North China plant is an example of widespread soil salinization in China. It is no coincidence that in China, as well as in Ukraine, the issue of plant adaptation to salinity and agriculture on saline soil is receiving increasing attention. genetic research and research in the field of plant ecological physiology are widely conducted as part of this issue. Against the backdrop of increasing salinity in China and Ukraine, questions are being raised about the feasibility of expanding the cultivation of salt tolerance crops. Questions are also being raised about the feasibility of expanding the cultivation of crops that are not halophytes, but can be adapted to saline conditions. Packing actually represent this second group of plants. However, these can only be done if we understand the mechanisms of response to and adaptation to salt stress inherent in packing. This indicates a relevance of our research topic for both Ukraine and China. We agree with Mr. Lubin's second point. We only have to clarify that we performed the volatility analysis in accordance with the generally accepted algorithm developed by the author of this analysis, Julian Lubin. This algorithm is decompressible in detail in his research paper and the research papers of his students. Therefore, in the thesis, we described the methodology of the volatility analysis quite briefly, with reference to the work that provide full information about the calculation algorithm used in the volatility analysis. We agree with all other content may be universal. Taking into account his suggestion, in the future, we will definitely pay a considerable attention to the implementation of our results in production and their inclusion in the system of technology for growing melon and representative of the general capability. Thank you again, universally.
Okay, thank you. Yuri Vasilievich, are you satisfied with the answers?
Yes, of course, thanks.
Okay, thank you. Continue. I invite an official opponent to speak, Oloykur Pisarenko, please.
Thank you. Review of the official opponent of PhD thesis of He Suntao. Once of the topic and the obtained result. The problem of increasing the area of soil on the air in complex in its causes and conservation. At the same time, the state of natural and artificial ecosystem will be primarily determined by the ability of living organism to adapt to saline conditions. Taking into account the peculiarities of the way of life and rules in ecosystem, the process of responding and adapting to soil salinity, which are realized by plants, are critical. For cultivated plants, this is also directly related to the issues of ensuring the efficiency of agriculture and the provided of food for family. In this regard, the Cucurbitaceae family and the genus Cucurbita no expression was representative have long been widely cultivated around the world due to the pleasure of a number of economical wild trends. In turn, the successful existence of representative of the genus Cucurbita in saline soil condition is possible only if they implement a set of adaptation mechanisms that cover different levels of organisms, starting with a modular node. The most significant science result obtained by the postgraduate personnel. The work is distinguished by the fact that in present the result of a comprehensive ecological, physiological, and genetic analysis of the response of Cucurbita genus representative to salt stress. He Sentao has implemented research covering different levels of living organization to logically summarize the results. He Sentao has studied and characterized in detail the effect of salt stress on the size and vitality of pumpkin plants, on the leading physiological process, the metabolism of essential organic substance, and on ion absorption and accumulation. The result of the analysis of the expressions of VRKU gain family revealed a number of important regularities in the formation of salt tolerance based on the process occurring at the genetic and molecular level. The scientific knowledge of the obtained result. He Sentao dissertation has a number of indicators of scientific knowledge that relate to both the methodological aspect of the study and the result obtained. It is worth noting that for the first time, He Sentao logically combined genetic and physiological study with scientific application to plant population ecology, namely the methodology of assessing plant vitality by zlobe. In addition, for the first time, there have been parent 12 VRKU family transcription factor in pumpkin in response to salt stress and analysis the phytogenetic relationship with the salt stress in detail. For the first time, the ecological entertain the transformation that pursued during the cultivated of cucurbitaceae using growing technology and the use of observable mycorrhiza fungi were investigated and clarified. The further development of the theoretical and practical principle of the use to salt-tolerant pumpkins, rootstock for cultivation and watermelon have been gained. The practical significance of the obtained result. The materials of the dissertation were covered in scientific publication and included in the curricula on the specific of 101 Ecologia at the Sumy National Agrarian University. The materials present in the dissertation are a base of research conducted at a high scientific and methodological level. The above use a wide token of science, knowledge, and research which combine generally accepted method, the Wade's approach to solving A scientific problem has proper statistical processing of result and they object entertain. Research result of He Sунтао present 12 scientific involved, 3 articles, Web of Science, 3 articles published Professor журналов Украина и 6 abstract of reports Science Conference. The work structure meet the establishment requirements. In consist of abstract, list a publisher's work on the dissertation, list a conversation in introduction, 3 sharpest conclusions and list of source of appendix. The introduction is according with the establishment requirements present the relevance of the shortest topic is novelly the list of tag, object, and method at research. In chapter 1, the scientific literature in the topic of the dissertation research and analysis. Chapter 2, the methodology of research is covered in divide. The chapter contains 4 subsections. 3. The result of the research and present. The section contains 5 subsections: the ecological, physiological, genetic aspect of adaptation and response on genus cucurbita to salt stress are described in detail. Conclusions summarize the results of the conducted research. The degree validity of research result, reality of data and their knowledge. The reliability of the research result present in the dissertation is based on logical, scientific base, the use of the complex theoretical and empirical research method. Experiment field and laboratory ecological, physiological, and laboratory genetic study were carried out chemical, spectrometric, fluorescent, morphometrics, and validity analysis were applied. The structure of the dissertation is logical and it corresponds to a assigned aim and tag defined for it. The dissertation is a complete scientific study. Total corresponds to the field of knowledge, then natural science, specifically ecological. Evaluation, the dissertation positively, the following wish and shortcoming should be noted. In chapter 1, subjects 1,3 is significantly larger than the other. To optimize the proportionality between the subjects, the literature review of genetic aspect of providing salt tolerance plants should have been present in a more concise format. 2. The paper does not characterize in sufficient detail the property of the pumpkins varieties covered by the study. For example, cultivate biome 112 was used in the study of pumpkin rootstock on the growing of grafted watermelon. 3. The information in figure 2,1 should have been present in the appendix. 4. Despite the fact that the work in well illustrated in it would be worthwhile to present 3 determination graph and models that would demonstrate the response of representative of the genome cucurbita to different level of salt stress. Reasons for the dissertation, taking into account that the state of agroecosystem and three productive larvae determined by the state of the rhizosphere, He Suntao should continue and expand research aimed to study the effect of arboreal mycorrhizae fungi in the state of plant genus Cucurbita and their agropopulation. General conclusion. The dissertation work of He Suntao, complete ecological, physiological, and genetic analysis of the response of Cucurbita genus representative to salt stress was submitted for defense to the specialized academic council of obtained the degree of Doctor of Philosophy in the field of knowledge, natural science, specific ecology. In terms of relevance, scientific and theoretical level, main result of validity, provision and result published in professional publication, notably in the formular and practical signation made the requirement of the ordered Ministry of Education and Science of Ukraine. Можно номера не называть. A base of the method about, I believe that the author of the dissertation, He Suntao, deserved to be awarded the science degree of Doctor of Philosophy in the specialty ecology on the field of knowledge, natural science. Thank you for your attention.
Thank you. He Suntao, you have a possibility to comment the remarks that have been made in the way of the official opinion. Please.
Okay. Dear Pavlo Viktorovich, thank you for your detailed and critical analysis of our work. Given that you are a well-known expert in the field of agriculture and green of the agricultural sector, all your comments are very important to us. We agree with them. Regarding the second comment, I can inform you later counted by me 100 times. It is a commercial variety development by the Pumpkin Research Group of Henan Institute of Science and Technology. It is famous for its high yield and good taste. I would like to emphasize once again that we agree with all your content. We will definitely work on the issue of generalizing the results of research on our chosen topic based on the construction of three dimensional graphs and models. Also, in accordance with your request, we will continue research amid at studying the rhizosphere and the impact of a vascular mycorrhizal fungi on the condition of cucurbita plants and their agriculture populations. Thank you again, Pavlo Viktorovich.
Thank you very much.
Thank you very much. Pavlo Viktorovich, are you satisfied with that?
I'm okay. Okay, thank you.
Thank you. We continue. According to the rules of work of the specialization academic council, we start the discussion of the He Suntao dissertation. I would like to emphasize that all members of the academic council have the right to take part in the discussion. So I ask everybody to express an opinion about this work.
Okay.
Please.
Thank you for the permission to take part in discussion. It is well known that soil salinity is a global challenge affecting the modern agricultural production worldwide. Therefore, studies aimed at clarifying the mechanism of plant response to this stressogenic factor are relevant. Pumpkin is considerably more tolerant to soil salinity and it is usually cultivated in arid and semi-arid regions. But for successful use of water, it's necessary to know mechanism of plant adaptation, of plant development, and its tolerance to such adverse factor. But features and nature of adaptation to this stressogenic factor to high level salinity remain studied insufficiently until now. Therefore, the relevance and importance of He Suntao research is indisputable. Thanks to various and different scientific approaches and methods which the author used for his investigations, in particular field, laboratory, fluorescence, physiological, ecological, vital analysis, and so on, the whole series of adaptation of pumpkin plants to soil stress is very vivid. In particular, it has been proved different mechanism related with photosynthesis and water exchange under saline conditions. Identification of expression of the WRKY gene family under soil stress condition was carried out. To my mind, it's very interesting part of He Suntao investigation was connected with the use of mycorrhizal and vascular fungi in cultivation of melon plants. That revealed some changes in metabolism contribute to increasing tolerance of plant to this stressogenic factor, I mean high level of salinity. The research conducted by the author extends our knowledge about nature of plant tolerance to soil salinity and makes a significant contribution to establishing the ecological and physiological nature of this phenomenon. I'd like to add some words about my experience communicating with He Suntao. He 4 years ago, he was a PhD student and studied in Ukraine, and we met with them, and I remember our today's applicant and as a very persistent, very successful and curious student. Based on the above, I'd like to express my support to the research, to the dissertation of He Suntao, and I'm sure that he deserves to be awarded the scientific degree of Master of Philosophy, Doctor of Philosophy, I'm sorry, in specialty 101 ecology, and I will vote for it. Thank you.
Thank you very much. Thank you. Please, your turn.
Thank you for letting me to say a few words about my impression. In general, I got a very positive impression from He Suntao's dissertation work. Their research topic is scientifically relevant and practically significant because it is a comprehensive study of the mechanism of resistance of agricultural plants to soil stress. I see that a lot of modern research methods were used in his works. For example, the identification and analysis of the expression of genes, and it has been demonstrated that salt stress affects indicates and characteristics related to the process of photosynthesis, water exchange, amino acid metabolism. And some classical methods were also used of determination morphometric parameters and vitality plans. Also, it has been demonstrated the influence of mycorrhiza fungi for the plant metabolism and their productivity. So He Sунтао showed a high level of theoretical training, deep knowledge of the object, subject, and methods of research, the ability to design and present his scientific work for definites. This applies to hard and soft skills of Doctor of Philosophy. Research results, which are certainly characterized by scientific novelty, were published in international scientific publications and can be used in practice. For that reason, I agree with the conclusions of the official opponents and reviewer that dissertation of Хе Сунтао, which was submitted for definites for the specialist and academic council in terms of structure and content, it corresponds to scientific research at the level of Doctor of Philosophy. In my opinion, also, Хе Сунтао deserves to be awarded the scientific degree of Doctor of Philosophy in the field of study 10 natural science, specialty 101 ecology, and I will vote for it.
Thank you. Professor Yuri Vasilievich, please.
Thank you. I will vote for this dissertation of Хе Сунтао, complex ecological, physiological, and genetic analysis of the response of cucurbita genus representatives to salt stress is relevant because it is designed to establish a mechanism for responding to the salt stress and adaptation to it, which are implemented by representatives of the genus cucurbita at different levels of organization, as well as environmental and physiological aspects of the formation of salt resistance, quantitative and quantitative characteristics of plants, grafting technology. I would like to note the practical significance of the result obtained in particularly the results obtained were used in the education process of specialties of 101 ecology at Sumy National Agrarian University, which is confirmed by the certificate. The work is written logically. The conclusions correspond of the objectives of the work. The conclusions are logical. In general, I believe that the dissertation meets the requirements for similar works at the author. Хе Сунтао deserves to the awarding the scientific degree of Doctor of Philosophy in the specialty of 101 ecology of the field of knowledge 10 natural science. Thanks.
Thank you. Auditor, please.
The work of distinguished by the fact that it presents the result of a comparative ecological, physiological, and genetic analysis of the response of cucurbita genus representative to salt stress. He Sунтао has implemented affected, research covered different levels of living, organization, and workers summarized to result. He Suntao has studied and characterized in detail the effect of soil stress on the site and object of curving the reliability in research result. The dissertation has a high level of science knowledge. Based on the mentality about, I believe that the author of the dissertation, He Suntao, deserved to be awarded the scientist degree, a doctor of philosophy in the specialty ecology and the field knowledge natural science. I will vote for this.
Okay, thank you very much. And for me, it's very interesting work. It's a very interesting science for Ukraine, for China, and I believe in He Suntao in future. And I will vote for this. Thank you. I invite to speak a scientist supervisor, Doctor of Biological Science, Professor Victoria Sudar. One minute, please. Okay, Victoria, please.
Dear Head of Commission, dear participants, thank you for the opportunity to speak. As already mentioned, He Suntao entered the PhD program at Summa National Agrarian University in 2018. Since then, I have been appointed as his supervisor to carry out research aimed at clarifying to features and patterns of response of chemical orbital representatives to salt stress and adaptation to it. Indeed, the subject of He Suntao's research is relevant, and the research itself is complex. They cover different levels of organization of living organisms and consider different means and aspects of the formation of plant salt tolerance. In my opinion, the research conducted by He Suntao made it possible to achieve the research goal and realize the objectives. The result obtained are of scientific novelty, as have already been noted by both reviewers and opponents. The results of the work have been covered in detail in a number of scientific papers and have passed required approbation. During the courses of his work, He Suntao proved to be a diligent and responsible scientist. He is proficient in modern methods in field physiology, genetic, and ecology research. He is able to set complex scientific tasks and achieve their implementation. So, as stated in my review, as a supervisor, I must reiterate that, in my opinion, He Suntao's dissertation is a completed scientific work that meets the requirements for works of this rank, and its author deserves to be awarded the degree of Doctor of Philosophy in the specialty 101 Ecology. Thank you for attention.
Thank you, Victoria. Does anyone else want to speak? Colleagues, dear colleagues? No?
No.
If there are no objections, so we stop our discussion. Ми переходимо на державну мову. Попередньо було розроблено проєкт рішення спеціалізованої вченої ради. Дозвольте його зачитати, шановні колеги. Спеціалізована вчена рада Сумського національного аграрного університету Міністерства освіти і науки міста Суми прийняла рішення про присудження ступеня доктора філософії в галузі знак 10 природничої науки на підставі природного захисту дисертації комплексний екологічний, фізіологічний та генетичний аналіз реагування представників роду кукурудзі та на сольовий стрес. complex ecological, physiological, and genetic analysis of the response of cucurbita genus representative to salt stress за спеціальністю складова екологія. Хе Сунтао, 1980 року народження, громадянин Китайської Народної Республіки, освіта вища. В 2010 році закінчив магістратуру в Фінляндському університеті за спеціальністю генетика та селекція рослин. Працює в Фінляндському інституті науки та технологій з 2011 по теперішній час. Навчання в аспірантурі Сумського національного аграрного університету з 6.11.2018 по 6.11.22 року. Дисертацію виконано у Сумському національному аграрному університеті Міністерства освіти і науки України. Науковий керівник Скляр Вікторія Григорівна, доктор біологічних наук, професор, завідувач кафедри екології та ботаніки Сумського національного аграрного університету. Здобувач Хе Сунтао має 12 наукових праць за темою дисертації, з них 6 статей у періодичних наукових виданнях інших держав та статей в наукових фахових виданнях України. Тому, колеги, якщо немає заперечень, питань, дозвольте перейти до процедури відкритого голосування. Прошу членів спеціалізованої вченої ради проголосувати і озвучити своє рішення щодо присудження Хе Сунтао ступеня доктора філософії в галузі 10 природничі науки за спеціальністю 101 екологія. Олена Олексіївна.
Я за.
Людмила Миколаївна.
Я за.
Юрій Васильович.
За.
Дякую. Павло Вікторович.
Я за.
Я також за. Голосую, тобто дасть за 5 членів, проти 0. На підставі результатів відкритого голосування спеціалізована вчена рада присуджує Хе Сунтао ступінь доктора філософії з галузі знань 10 природничі науки за спеціальністю 10 екологія. Шановні колеги, чи є зауваження щодо рішення спеціалізованої чи взагалі процедури проведення спеціалізованої вченої ради? Нема?
Немає, все
Немає.
чудово.
Я щиро вам дякую. І Хе Сунтао, congratulations. Your finish report, please.
Thank you.
Thank you very much.
Ні, не переписано.
Окей, sorry, your finish report, please.
Можемо сказати.
I thank all the members of the specialized academic council for their detailed and critical analysis of our work, for their comments and suggestions, and for the positive the decision to award me the degree of Doctor of Philosophy in Ecology, and also thank all the teachers of Sumi National Agrarian University, and especially the Faculty of Agriculture, Technology and Nature Management, and the Department of Ecology and Botany, who work with me during my graduate studies. I'm also grateful to my supervisor for her help and support while studying for my PhD and writing my dissertation. I gained an important result and experience that will definitely be useful to me in my future research activities. Thank you all once again.
Thank you.
Thank you very much.
Congratulations, Хе Сунтао.
You are the best. Thank you, Хе Сунтао. На цьому, шановні колеги, засідання спеціалізованої вченої ради вважається закрите. Дякую щиро всім за роботу. Всім мирного неба. На все добре.
Дякую.
До побачення.
Дякуємо. Навзаєм.
До побачення.